Okay, thanks, Sean. Welcome everyone to the LIMS seminar. Hopefully everyone can hear okay. The structure we hoped is going to be informal. We have presenters that are going to present technical information and just use a live view of LIMS. So we're going to actually use a live view to present how to do some tasks on LIMS. LIMS is a laboratory information management system, and it's a generic term. A lot of large labs, commercial labs have a LIMS. There are commercial off-the-shelf. Our LIMS is custom-made for the KSSL. It's developed by here at the lab over several years, and it's done specifically to meet our purposes. It's used to log in samples and metadata, enter the associated metadata, assign analysis, enable processing and preparations of samples, prioritize projects, and make up batches. It also records the instrument information, results of analysis, does calculations, and records and distributes or makes available the data. So essentially all of the lab operations are dependent on LIMS. Probably for you as liaisons, the most common applications that you're gonna use LIMS for are determining the status and progress of analytical projects, and that includes verifying that samples have been received by the lab and identifying or inventorying how many samples there are in a project, what kind of samples they are, and looking at analysis selection and metadata associated with those samples. You're also going to use it to determine the progress of analysis. That's probably one of the most frequent inquiries you're going to get is what's the progress. So we're going to go over that, how to evaluate the progress. You can also access for projects that aren't complete. You can access and distribute the partial information. The analysis that are completed can be accessed and distributed to the submitters. You also will be able to retrieve and review completed data, request reruns, and then evaluate related data. So a lot of the functions that LIMS does you won't need or do. So there are some features in LIMS that will be there, but you probably won't be actively using a lot of them associated with the actual operation of the lab. Okay, today Rick Nesser and Scarlett Bailey will be giving the technical presentations. Rick is the person who maintains our LIMS, including the upgrading and revising of the code, revising calculations, adding new instruments, and making changes to procedure. And Rick can also investigate any glitches. He can also produce custom output or do queries that aren't necessarily covered by the standard reports that we're going to talk about. So Rick is our person that maintains the software in the LIMS system. Scarlett Bailey is the person who initiates projects, enters analysis requests and metadata, and moves the projects through the levels of progress of the LIMS system. She facilitates the review of completed data and distributes completed projects and queries completed data. She is the person who probably you will interface with for common questions or data problems or anything that you would have, and also to access some of the reports. So with that, I'm going to let Rick start and kind of give an overview of LIMS and what you're going to see when you actually access LIMS. So.
Well, thank you, Steve. I am Rick Nesser. I worked here for a long time, helped develop our KSSL LIMS product. As liaisons, you will be using LIMS as Steve had mentioned in performing your duties. Hopefully, all of you have had an opportunity to request that the KSSL LIMS program be installed on your personal laptops or workstations. It is CCE certified and has to be anytime it is modified. To prove it, you should be able to see a KSSL LIMS icon. Screen is shared, right?
Yep.
Okay. So you should be able to see that. It is my duty to ensure that KSSL LIMS knows who you are. And so I have to modify a table in the LIMS database that will tell LIMS about you. To actually start up LIMS, you should just be able to double-click on this icon. And what it will do is connect up to our host, which is a database server located in Lincoln, Nebraska. For that reason, you can only use LIMS if you're connected through the USDA backbone network. That means that anyone sitting remotely can VPN in if they're working from home or anything like that. As long as they're connected to the USDA backbone, they should be able to connect to the LIMS server. And the proof that we had success is the fact that we are now looking at the LIMS main menu. If we had trouble connecting, you would have gotten a separate window saying that there's a problem with connectivity, and in which you would call me to try to rectify that. And you can see down here in the bottom that it does know that Sean McVeigh is the one who's logged in, and there's the date that you logged in and the host, which is the server name. So I'm going to work through the command bar up here from left to right to explain a little bit about what features LIMS has. And I'll just simply briefly cover them. The application command helps control the window itself. There's a toolbar which has icons which match these commands. So if I wanted to get more space on the window, I could click this and the toolbar will vanish. Similarly, the status bar at the bottom, if I click on this, will make it go away. So it gives me more room on my window. We have unit conversion, which means that if you wanted to be able to convert units of measure, if you're typing in values that maybe someone gave them to you in inches and you wanted to convert them to centimeters, you can simply put in the inches, like 25 inches, and immediately it calculates how many centimeters that is. The sample command is mainly used by the technicians we have in the laboratory who work primarily in the basement. They're the ones who receive the samples that come in from sample submitters. They're the ones who log in those samples into the laboratory information management system. And that's what this command is for, this submission. You will typically not get in there. Once samples have been submitted to the laboratory, then paperwork which came with them will usually go upstairs to the laboratory office, in which Scarlett Bailey will receive those and do further work. The technician lead is Michelle Ettman, and she may contact you in regards to sample submissions if there's any issues. So once samples have been put into a project and the analysis scheme has been entered in, then technicians in the basement will open up this preparation window, and they'll know what they need to do to take the soil samples and make preparations so that they can be analyzed. The disposition is used by technicians to take care of the preparations, either make them available for analysis or literally throw them away when the time is done for using those preparations, or maybe even put into permanent storage. The project command is for when you want to manage the project as a whole. That is, identifying who the contacts are, attaching spreadsheets or any other documents to the project, assigning analyses. And you can go in here and look at things, but typically, again, you will not be actually altering anything about the project. The analysis command is after a project has been given an analysis schema or scheme, then as I said before, the technicians get the idea that they need to prepare these samples for analysis.
Yeah.
And then once the analyses have been, or the preparations have been prepared and made available, then the technicians will get a list of, kind of like a to-do list. They'll see that particular preparations are ready to be analyzed, and certain technicians will create batches of samples that will be analyzed as a whole. Then, later on, when it's assigned to them, technicians will come and literally analyze the batch of samples and enter the data. And that's what this data acquisition window is all about. Typically, you will not go into there. Now, the report window is what I'm going to spend more time on because this is the one thing that you will be using the most, which is to generate reports. Scarlett will spend more time with you on this, identifying which reports will be valuable to you based on the projects that are going to be assigned to you. But one thing I did want to mention is that there's a wide variety of reports that are listed over here with check boxes. And I just simply wanted to explain this interface a little bit. And all the reports are arranged by a section, either a laboratory section like biology, chemistry, particle size, or there are general reports that you can create, and they're usually based upon a project. So if I wanted to see the project analysis status of one of my projects, I can scroll over here. If I know the name of it, great, but if not, I can then sort this list based upon the coordinator name, and if I'm in this list, then I can find my name and find the project I'm looking for. So I'll pick on I guess I can choose Larry West. No. How about Mike Wilson? We'll pick on him. So if I have a project that belongs to Mike Wilson, say this one, I think, then I can add this to a list of selected projects. I can add as many as I want into this little window. And then when I click on Open Reports, it will open up a cross product of all the projects I've selected versus all the reports I've selected. In general, LIMS uses a web browser. That's what this window is. It's like a web browser. And the important thing to note about that is there will be a URL. Now the URL is kind of cryptic, and it's really relevant to the web server that we have. So the beauty of having this URL available to you is you can copy and paste it into your local web browser on your desktop, and it would be available to you anytime. But this web browser has been fitted to be able to do things like if you are running a report, you want to attach it to a special project, you can do that by clicking this command. You can print them out if you like, or send them through email to somebody you want to send them to. And because it's a web browser, sometimes these reports may time out if you're asking it to do a lot of work, and it will show up in this window. If the timeout happens, what you should do is go ahead and immediately refresh it, because chances are it'll pick up and redo the report, and the next time not time out. Just a hint there. So that's really all I want to say about the report. Under Tools, you have a number of parts here that are mainly used by the technicians to support the data acquisition. So I don't really want to say too much about that. And this is all I really wanted to say about the LIMS interface. As Steve said, if there's any problems with using it, you get windows popping up that say there are errors, then I want you to either take a screen capture of that and email to me, or you can use Skype and talk with me about what's going on, and we'll try to get your problems rectified as soon as possible. I'm going to turn the meeting back, over to Scarlett, and then she can continue talking about what parts of this LIMS you're going to be using.
Thank you, Rick. For those of you that may not know me, my name is Scarlett Bailey. I'm a data management specialist for the Kellogg Cell Survey Laboratory, kind of a project manager role as well. So I'm the person that validates projects when they come into the lab and kind of keeps an eye on them and keeps them moving. Like Rick talked about, one of the Windows menus that you'll be using is this project menu. Head-ons and samples are organized by project in LIMS, so you'll be searching for a specific project most of the time when you're in here, not just a particular sample of interest. From this menu, you go to the laboratory listing, and then you get this window here, which shows you a listing of all the projects that are going on in the laboratory. The window defaults right now to just showing projects that are considered active. That is, projects that have a status of in progress or validated or review, and I'll talk about what that means in a minute. But if you want to see more than just in progress projects, you would toggle one of these boxes here to look for projects that were in a pre-submission status or even completed projects. From this window, I'll explain each of the columns. This priority column is the ranking number that is set for the particular project. This number is set after the project has been validated. It's usually entered by me or by Steve, and it can be adjusted at any time as needed based on laboratory priorities. So just because it's at the bottom of the list doesn't mean that can't change. We always are switching things up to meet certain needs, certain deadlines, whatever we need to do. And that priority number dictates where samples will appear on batch creation lists. So a project with a priority number of 2 is going to show up higher on the list of samples to create a batch from than priority 5. Oh, yeah, you can't see all these projects we're working on. Here you go. Here are some priority project numbers. The next column is the lab project number. This number is automatically generated by LIMS and assigned. The next column over is the project name. This is the name that's given to the project, usually by the submitter. They tell us what that is and we enter it in. The status column, this will tell you what status the project is currently in. If you see a status of pre-submission, that means that the samples have been logged in. They've been received by KSSL and logged in. Maybe some information has been entered, but it hasn't been set in motion yet. Another status that you will see is validated, like this last one down here, validated. That means that I've entered all the information about the project, entered the analysis request, the preparation request, sent out the report to the project submitter, and now it's ready to go. It's just awaiting either processing or analysis. A status of in progress means that the samples are currently being analyzed. They're working through the lab. A status of review means that the project has been completed, but it's just going through, the quality control kind of review by liaisons. And a completed status means that all of the analyses have been completed. We've closed out the project, and if it is a public project, at that point, the data gets published to the Lab Data Mart website and is available for download by any outside parties. And that's only for public projects. We can mark projects as KSSL only, which means they wouldn't be published at the end of the project cycle. Another call? Yes?
There was a status of validated, but we don't have that
right.
filter.
Validated is considered under active. Active is in progress and validated projects. Good question.
And do we really have projects going back to 2006?
That are currently being worked on? Yes. We might be doing some additional testing or found some reruns. So yeah, we have projects from all years going all the time. The next column you'll see is a due date. If there is a due date, the row gets highlighted yellow. Sometimes we'll have projects come in that are like for conferences or field trips or soil judging contests, and the project submitter needs their data by a certain date. So we'll add a due date here to kind of help remind us that we need to get this done by a certain time, that somebody is waiting for it. There's no like automated reminders or anything that come with this due date. It's not like on October 31st, I'm going to get a thing that says, hey, this project is due tomorrow. But it's just to help you kind of keep an eye on when it will be due. The next is the number of layers that are in that project, the number of samples that have been submitted in that project, the date that the project was received and logged into the LIMS system. And if, for instance, somebody were to send additional samples for a project later on, the date that shows here is going to be the most recent date that samples were logged in. So sometimes we'll get additional samples that go with older projects. So it's going to clear out the original logged in date and put the most recent logged in date there. The last thing is the coordinator, who the project coordinator is. It's typically the liaison for the project area, but sometimes for special topic projects, they'll be working with somebody specifically from the lab, or will have sent their request in specifically through that person. You can sort this window using any of these column headings here, alphabetically, by due date, layers, if you want to group all your projects, by person. You can also filter just to see certain projects from certain fiscal years. If you know the project name or part of it, you can start typing in this little window. I typed in any to filter down to the Nebraska projects that are in progress. What else? There's two little buttons here that will help you identify a project if maybe you don't know what the project name is. Find project will bring up a little window like this. This would be like if you know the name of the project to search on. Maybe you want to look just for projects that you are listed as a coordinator on. Maybe you know who the project submitter is. So like Rob had sent in a whole bunch of projects. So if I put in his name and search, then it shows me the 2 active projects from fiscal year 2019 that were submitted by Rob that are in progress here. So this is one way that you can find a project if you don't have all the information about it. Otherwise, you can also use this Find Sample window. If you know like the sample ID, maybe you know when it came into the lab, or somebody says, hey, I think we sent these in May 2018. If you know the layer number, Headon number, user Headon ID, like say somebody said, hey, I think I sent some 2017 projects in from Wisconsin, and you have no idea what project they're in. You just know somebody's looking for Wisconsin 2017 Headons. You can use the wildcard, kind of type in the information that you know, and then you'll get your results down below, and then that can kind of help you get to the project of interest. Like, oh, it's this project. So I select it, and here it is right here in my window.
And you can also, in the lab project name, start typing it in, and it will, for instance, if you know it from Nebraska, you can do NE, and then it will show those, so that's handy sometimes.
Oh, I'm on the completed. Yep. Are there any questions about this so far? Am I going too fast?
Well, who assigns the priority? Because some entries have one and some don't. It doesn't mean anything.
It does mean something. Steve and I will assign the priority. For these projects that don't have a priority number, you'll notice that they're mainly older projects. It means that usually somebody from the lab, we brought them back into progress to do some additional testing. There's not a priority number on it because maybe it's an older completed project and we're just doing some additional things. And without a priority number, it just shows up at the bottom of the batch creation list. So, which is fine because when we're doing this, we usually know to look for those samples on the list. So ones with a priority will be at the top, ones without will be at the bottom.
Generally, they're added as they arrive to the bottom of the list, and they're just as received.
Right. The priority number is generally kind of sequential as they are received or as they're validated. But like I said, we can adjust that at any time. If there's one with a higher due date or a higher priority, we can change the number whenever as we need to.
And the due date column is, a lot of times that refers to something like for instance, we did some samples for the NCSS meetings that had a definite time that they needed to show for them, so we put a date in there to make sure those were done on a time.
Yep.
So a lot of times that's some special meetings or presentations or something like that. Those
right. Make sure we get them done in time.
But those as a liaison, if there is something like that, then you should probably, and you know it, you should probably communicate. A lot of times a submitter will, but you could communicate that to Scarlett that there is a specific overriding need for those, and she can check on those. And then you would check, as time goes by, you can go into LIMS and check that project, check progress on it, and also kind of as a backup, because like Scarlett said, there's nothing that's just an indicator. So you can kind of ride herd on those types of projects and communicate with Scarlett or somebody in the lab if it doesn't seem like it's going to work by that time, and to make sure that gets done.
Yep. Okay, so I'm going to go into the project view. I'm going to open up a project here as an example and show this project view window. And what you see here in this middle section is like a nested tree view of all of the Headons, sites, Headons, layers, and samples that are logged into the project. And you can use the little plus or minus to collapse or expand as needed. Got the project name, here's a site ID, here's a Headon, it's got the particle control section here, the sampled as taxonomy, and then each of the layers, and then each of the samples that are associated with that layer. You'll notice some color coding here. The layers that are blue, if it's highlighted blue, that means that it is within the particle control section. Like you'll see, it's 2 to 102 centimeters. So these 3 are highlighted blue. These ones are not in the particle control section, so they're black. And then you'll see these samples here are highlighted in red. And the red indicates that these are regulated samples. They're APHIS regulated samples, contaminated in some way. And here's a little, oops. Down at the bottom, you can see the key blue layers with Headon control sections, red for regulated samples. This is kind of a good summary view to when you're trying to figure out maybe an identifier or see what was submitted for a project. Using this tree view is really helpful. And then there are some navigation buttons here on the side. If you open up the project, just kind of gives a little extra information about the project. This is where we would designate it as public or private if we needed to. What office it came in from, the project name, submitter. If there was a due date, we would enter it here. I wanted to show these buttons at the bottom, this attachments button. This is a place where you can upload files and attachments to keep with the particular project. We have a hard copy project folder also that you could, print and keep it in the project folder, which is stored at my desk, but you can also use this attachments button to save it electronically with the project. I've been trying to use this more in recent times. On this particular project, like I've got the sample submission sheets straight from the submitter attached here. So then if you ever needed to reference those, you could do it through LIMS rather than, coming to look for the hard copy project folder. So I would encourage you to take advantage of this attachments. I believe you can attach pretty much any type of file, any file type. I don't think there are size limitations or anything. You can delete things, so just make sure you don't delete somebody else's stuff. But that's a good thing. The other thing I wanted to point to was this notes section here. Anyone can write notes. This is a good place to, again, keep project information electronically rather than in the hard copy paper folder. If you have questions about a project, like maybe some layers were missing data or any kind of question, this is a good place to go to when you're investigating those problems, because a lot of times technicians will write notes here or processing will write notes here. Like sometimes samples may get lost in shipping or open in the bag. Then they would indicate that here. Maybe the clods were too sandy for analysis, were falling apart. The lead physics technician will make a note here. So you can kind of see what was going on. Yes, Patty.
Scarlett, this is a place too that, let's say that you request a suite of analysis and you're getting your report and you go, well, how come they never did this? You'll have some empty spaces. How come you never did this? If you look in the notes, we try to make notes where like maybe the sample ran out or there wasn't enough to do, or maybe it was a problem sample where New Zealand couldn't be done because 5 grams would soak it all up. So you can look in there if you're like missing data and you don't know why, there may be a note there. Sorry.
Nope, perfect.
Viewable? Can you use an attachment? Can you view them? Can you open it up and read it?
Yes. Well, it'll show up right here. There's also a report that you can run that summarizes them all, runs them all together. But like if I wanted to add a note, you'd use this little thing right here, new note. Oh, it says Sean McVeigh. LIMS testing. I am writing a note about samples. And then, do I need to check the report box? If I check the report box, then it shows up on the output. Remind me, and I'll show that when I get to the report section.
And the same question for attachments. If you attach something,
those, it's not indicated anywhere else to see that there are attachments. Like you can't run a report that'll just spit out all the attachments or download them all as a zip file or something.
That's how you get them back. Like if I put an attachment in there and I wanted to get it back, I have to.
Yep, go to the project button, open up the attachments. And this little box, see how it has like a little gray check? That's your clue that there is an attachment there. Same with notes. If there were no notes, if there were no attachments, it would look like the contacts square.
I'd like to say that if you open up that attachment window, you can double click on any of those and it will open them up.
It will.
Yep. Yeah, it will open them up in the proper
software.
Right. Sorry,
Like
sorry if
that
I
one.
misunderstood the question.
Well, I don't know if that was the question. He said, can you get them back? Well, this way you can open it and then you can save it to somewhere on your computer.
Those relevant answers.
Okay, good. So yeah, I really want to encourage people to use this project attachments button when you can. It's nice to have things stored electronically and be able to access them from everywhere. So like I said, I'm trying to use that more, and so I hope that you guys will use it also.
And that sample submission sheet could be useful too. It's the original request from the submitter that gives the list of samples and all of the analysis they requested, and also that's a good source of reference. They should, when they submit the sample, that's emailed, and they're supposed to include the liaison on that email too. So you should get them at that stage as a liaison. A lot of times they neglect to do that,
or they send them just to me.
Yeah. So that's kind of an ongoing thing to remind them to do that. But that kind of lets you know that they're submitting a project.
Okay, that's the project. And I'm just going to blow through these. You won't be using them too much. This is the site navigation button for each site in the project. Has the geography information, MLRA, lat long if it was entered or came in from NASIS. Same for PEDONs. Here's the PEDONs from the project, control sections, taxonomy, layers view, just straight up list of all the layers, horizonation depth, texture. This is where I would enter in the texture for the layers. And then also a samples listing with all the sample IDs, the type, like bulk and field core, weights, and if the sample is regulated or non-regulated. And then these two buttons, I'll just mention, this analysis button is what I use to request analysis against the project, and same with the preparation. When I need to request that processing makes preparations for a project, these are where I would go to do that. And this notes button just takes you to the same place as the one in the project. It's just another way to get to it. Notes. Okay, any questions about this project view screen, how you might use it, why you might use it? Okay, let's talk about reports then. The next toolbar item that you will use a lot for your duties is the reports. This generate reports here. Rick kind of talked about it a little bit. You're going to be mostly using reports in the general section, but feel free to look at any of the other reports that may be of interest to you. I'm only going to talk about some reports in the general section. Let's see. So, like Rick said, you can sort this list to look for a project. You could type it in if you know what it is. The same find project and find sample buttons that I demoed earlier are here. Sort by, filter by fiscal year. And again, this view right now only shows projects that are active, in progress, validated review. So if you are looking for an older project, make sure you toggle this little box here, include completed projects. So sometimes you'll be looking for a project. I can't think of a completed one right now. And this whole thing will be blank, and you say, well, where is it? And then when you toggle this include completed projects, then you'll see the older ones there. Okay, let's see. I'm going to go back to that project I was using before. So I just started typing in the number because I know what it is. And I'm going to double click right here to send it up to the selected projects section. Like Rick noted, you could add as many projects here as you wanted. I'm just going to run it one at a time. The first report that you should be familiar with is this project analysis list. I sometimes call this the project plan. I'm going to open it up. This report shows all the sites and PED-ons for the project, identifiers, PED-on classification, all the layers, and then the analyses that were requested on the project and on which layer. And then so you can see each of these breaks is a PED-on, each of these lines is a layer. If it has a number in it under the column, it was requested. And then there's a summary down at the bottom showing all the analyses that will be run, how many samples from that project are getting that analyses, the preparations that will be created for the project, and then also the number of samples that were submitted. So this project had 41 bulk samples submitted, 12 field core samples for a total of 53 samples in the project. When this report comes, I will send it to the project submitter and the project coordinator at the beginning. After I validate the project, send it to them to make sure to have them review it, make sure I didn't miss any analyses requests, make sure that it's correct. And also I'll send it to the liaison project coordinator at that time to do the same thing. Look at it, make sure all the requests make sense for the soils, for the samples, compare with that original analysis request spreadsheet to, again, make sure didn't leave anything off or add anything that wasn't supposed to be there. Just make sure the project's ready to go so that we can get it in the oven, get it processed, and start analyzing it. So this, go ahead.
And is that, there are some things that may not be, for instance, if we usually don't do X-ray diffraction on A horizons because the organic matter interferes and you don't get anything. So if they had requested that, it might not be specified on here, so there may be questions like that, you know, as to why that wasn't selected.
Mm-hmm. So this is a good overall summary of what the project plan is, what's requested, what we're doing with it.
And we're working on a kind of a supplement to the analysis request sheet that enumerates a lot of those screens that will go with the sheet. So hopefully when we get that out, that will explain a lot of those kind of screening criteria that we may have for some analyses. Another report that you may use to view like sample numbers, layer numbers, that kind of thing is this project samples list. It gives an output of all of the sites, PED-ons, layers of the project, including the natural keys, and this report has the sample IDs on it, whereas the project plan only has the layer natural key on it. This report can be used to get from a sample ID to a natural key to go between the two.
Question.
Yes.
Natural key, is that what ends up being the lab sample number when it gets exported externally? I just wanted to double check that because I'm always confused.
Like in NRCS?
Yeah, well.
In NRCS, the PED-on natural key, this number right here is what's considered the lab PED-on number.
And that's what links it. But
and that's what links it.
Repository, then it will have the lab sample number. Is that what it's labeled as if you do the query on the website?
Yes.
Okay.
Yeah, this is Rick. That is in NRCS would be called the PED Lab SAMP-NUM column of the PED-on table. And the other natural key down below is the under the pH sample table analysis, and it's known as the lab samp num column.
Okay.
Yes.
Yes, but I just wanted to point out this report as a good way to go between sample ID back to natural key, because sometimes that can get confusing or cumbersome. And anyway, this is just a nice summary of everything that was submitted, and then also by sample type. So I can see, this layer had both bulk and some other type of sample. I think in this case they were field cores.
What was that report called?
Project samples list. Project samples list. Okay, the main one that you're probably going to want to use, this is to check status of a project. I'm going to switch to a different project here really quick. To the Montana project. Okay, I'm just going to open it up. So if you want to check how far along a project is, where it's at in moving through the lab, you would use this report here, the, project analysis status report. Right here it lists the analyses that are going to be done for the project on the row. And then each of these columns I'll explain. This requested column, that's the number of samples for the project that were requested for this particular analyses. So 115 samples for quad, 85 for calcium carbonate, 86 for CEC. The next column over is not available. That's the number of samples that have an analysis request on them that still hasn't been fulfilled, and the samples are also not available yet for analyses. So that would mean like they're still processing it and they haven't brought it upstairs and made it available yet. Or like we used up the sample before we got to this test and we can no longer do that test because we used all of the fine grind or something. With the rerun column, the number of samples that have been requested for reanalysis. The number to be scheduled is the number of samples that have been requested but are not yet in a batch. The number in progress is the samples that have been put into a batch for analysis. So like on the CEC line, you see there's one that's waiting to be added to a batch, but all 85 of them are already in batches and are at some stage of analyses. Next column over is not required. That is the number of samples that didn't need the particular analysis based on some criteria. Like it had too high or too low of a pH. The organic content was too high. Didn't effervesce. So like they requested gypsum on 85 of the layers. Well, 17 of them didn't meet the cutoff for our gypsum testing. So a lab technician, when they were creating the batch, marked them as not required. We don't need to do these based on some other criteria. Did I say that correctly? Does that sound good? Okay. The next column over is abandoned. These are the number of samples where the analysis was unrequested for a reason other than not required. The difference between the two, not required is it was marked not needed based on a criteria. Abandoned means we took it off for some other reason. Either I accidentally added it and I didn't mean to, or we ran out of the sample before we could do it, so without abandoning it, it would just hang out in to be scheduled forever. So that means just forget about it. And then the last column is the number of samples that have been completed for the analyses. So like in this example on the salt predict line, 87 were requested, 46 are still being worked on, and 41 have been finished. So if I add up the number of the finished ones, the number of in progress, I get to the number that were requested. Last thing to note is this total percent completed down here. It's not always perfect. Sometimes it won't hit 100%, it'll hit 99%, but everything will be done. Sometimes if there are reruns, it might go over 100% and show 102% completed, but it's still a pretty good idea of how far along the project is, kind of good general idea. In this case, I see 4%, so I know the project is just getting started. Down below, there's some other additional information. It'll tell you the batch ID that samples from this particular project appear in. So I know that for the CECs, they appear in these 1, 2, 3, 4 batches so far, anyway. It'll give you the list that the date that the batch was created, and then also the date that the batch was marked complete. So like for this salt predict, the batch was created on the 4th, and it was completed today. And then also just a history of the status down here, the dates that the status is changed. But you're mainly going to be concerned with this top section here, this progress indicator. Any questions on this report or reading it, how you might use it?
Charlotte, the completed date on the bottom there,
yes.
Does that show, like, I validate something, so sometimes I might complete it back and I go back into it, complete it again. Does it show multiple completion dates there, or is it just the last?
I think so. I think if you uncomplete and complete it, it'll show you each date. Is that correct, Rick?
I think
I don't remember.
That's the latest one.
It's the latest one on this report.
Yeah, it's the latest. If you put in like a sample number and put find batch, it'll give you both completed dates for that.
So I think it's the most recent completed date that appears on this screen. If somebody were to go back into it.
Could you go over one more time what's the difference between not available and in progress?
Not available means that there's a request, but that the preparation that's needed for that request has not yet been made available for analyses. So like on this one, I see there's 41 waiting for calcium carbonate. Well, why are these 41 samples not available? Calcium carbonate uses a fine grind preparation. The fine grind preparation can take a little bit more time. So I would say 41 of these fine grinds are still being made downstairs right now, have not been put into a box and sent upstairs. So that's what the not available is. And then in progress means that the sample has been added to a batch. It's either just in a batch awaiting analysis, it's being analyzed, or it's been analyzed but not yet marked completed. Good? Okay, that's a big one. When people want to know about the status, that should be the first place you go.
And that would be something, if there's something that's been hanging out in one of those columns for, I mean, you check it several times and it keeps showing up as not available or something, then you could ask Scarlett.
Right.
She could investigate exactly what the problem is. And at some point it might be, okay, well, there's just not enough to make the fine grinds, and that might, so then it might be shifted to abandon, and that would mean that won't show up on your final data.
Correct. We only have a couple minutes, so I'm going to go real quick. There's this NASA's Headon description report. This would save you some time from going into the NASA's program to run the report against Headons. If you know what project and such it is, just use that. It will only work if I have added that lab Headon number to the Headon in NASA's, but usually I do that during the project validation, so that's taken care of pretty early on. But yeah, this would save you some time from opening up NASA's, querying for the Headon, running the report, opening the report. It's just, you can do it right from LIMS. And alternatively, if you were, say you were only interested in like one of the Headons from this project, you can use this Headons tab here and select just the Headon that you want to see. Double click, see how it's over here in the selected. I'm going to remove the project one because I don't care. Open the report, and I should have just the description for this one Headon. Only this Headon, not the entire project. And that can come in handy sometimes when you don't want to print everything, or if it's a huge project. Oops. You can just switch over to the Headons. And so you can, yeah, that's how you get there. The other thing The primary characterization report, you should be familiar with this. There's also the supplementary characterization report, the ANDIC properties report, the taxonomy report, which also show up on the Lab Data Mart website. Those are also here. I'm just going to show the primary characterization report. You can run this at any time against the project. It's only going to show the validated data from the project so far. So like if you run it really early on in the project, it'll probably look pretty blank because we haven't done most things. But as the project gets going, it'll fill in.
In order for those to be filled in, the project status would have to be set to what?
Any status.
Yeah, it's probably going to be active or in progress.
Yeah.
But that's the access that you have as a liaison that the submitters don't. This will not show up at the website until it's completed, until it goes completed. Then the submitters can go online and do it. But meanwhile, you can, if you see that it's 50% done and you want to generate a partial data to give to them, then you can do that through LIMS at any time, even if it's still in progress.
Are there any other reports that will give us tabular data rather than HTML?
There are, the question was, are there any reports that output the data in a tabular format? That's a request that we've been getting a lot more. So Rick has worked up a couple of provisional reports that download as CSV files. We're still kind of figuring these out, working out the kinks to make them work as intended.
Why don't you show one of those?
I will surely show one of these. The primary tier download, it'll prepare a zip file with CSV files from each tier of the primary characterization report. So hopefully it'll go quick. When it's done, it'll come up with like a little link to download a zip file if you use the primary tier. And what I say, it'll have a CSV for each tier, so like the bulk density tier, the PSDA tier, carbon and extractions tier, and then also a data dictionary file. The other one here, the single table download, it does the same, uses the primary characterization data tiers, but sticks it all in one file instead of different files for each tier.
And that one will work faster than the one that we've asked for, unfortunately.
Let's see. So yes, these are provisional. They're under construction, but yes, we're trying to meet that tabular data need for people. Hopefully it'll run. Okay, so then tab delimited laboratory data file was created. Makes a text file. If I click on this one, okay, so there's all the data in a tabular format. What I would do, I would save this, save as, and then, import it into whatever spreadsheet thing I was going to use. To get back to the original window, I just right-clicked on it and clicked back. And then this file down here is the data dictionary that goes with it. It explains each of the column headings, analyte code, name, description, but it goes with this file, explains the laboratory data file.
That makes my heart happy.
Yes, it's good. It's very good. A long time coming.
Just for you, Scott.
We sure did.
Just for you. We want you to be happy.
Yes. So, yeah.
And those other reports, like the ANDIC report, are kind of underutilized, but they're very helpful in that they say, if you look like for the 1/2 iron plus aluminum extractable that you need for ANDIC, it's already calculated in there. So those are, look at the, get your folks to look at those. They're useful, and some people don't even know they're there.
The last thing I wanted to say about this thing is, since it is a web page like Rick noted, you can Control-C to find things. So like say you're looking for a particular Headon or a particular layer, you can use that there. To save it, file, save as, and I would save it as an HTML file, rename it and save it as an HTML file. If you are going to print this out, I highly recommend doing print preview. I don't know about your computer, but mine defaults to the up and down portrait mode. And I like to look at these in landscape mode, so you'll want to change it over. Also, you can see this is a huge project that will print out 93 pages. So before you hit that print button, maybe do the print preview and make sure you're not printing out a 200-page report. Maybe just get to what you need or whatever. But yes, I highly recommend utilizing this print preview. I don't know how many times I've printed out a primary characterization report in portrait and then been real mad about it. So use that. The last things I wanted to say, we've kind of already talked about. If you have any questions, comments, or issues, if you're having like major problems with the application, if you're getting error messages, if it's not opening up right, or if you have any questions about the reports, you're interested in a custom report or changing one of the reports that's already in there, contact Rick Nesser. Basically, for most other questions, feel free to send them my way. Questions about just general using LIMS, how do I find XYZ, can you show me that again? Or if you have questions about project status or certain batches or samples or those types of things. And if I don't know, I will triage and kick it up to Rick. So now I guess open it up for questions, or is there anything that Rick or Steve would like to add?
Yeah, that's good. You have done a good job. So.
Thank you.
Okay, questions. Do you have any questions from anybody on the phone? Okay.
Nothing in the chat window. Anybody online have any questions, comments, concerns?
Yeah. Like Sean said, this is going to be available on the
our
YouTube
center's YouTube channel.
Okay.
Right.
Okay. And then also, meanwhile, you know, like Scarlett said, feel free to contact her or Rick or myself or even any of the individual technicians that might be able to help you. You know, appreciate everyone's attention.
Yes, thank you for your time, and I hope this was informative and you learned something about LIMS that you didn't know before.